Hello, everyone. My name is Diana Wall, and I'm the director of the School of Global Environmental Sustainability at Colorado State University, and the chairperson of this National Academy's committee, study committee. In my role as chair of this study and as the moderator of this committee meeting, I'd like to welcome everyone here on Zoom, those of you watching the live stream, and all who may watch the recording later. The task being undertaken by this National Academies of Sciences, Engineering, and Medicine committee is to explore the linkages between soil health and human health. The committee's full statement of task can be found on the study website, which is available at the link under Project to the right of the embedded video of this meeting. I'd like to give you some sense of what's coming in today's meeting. I have a few housekeeping comments to make, and then I will turn things over to Carol Laney, the study director with the National Academies, to provide a brief overview of the study process and what we are doing. Following her presentation, I'll introduce our speaker for today. To start, I want to note that this is an open, on-the-record session. Interested individuals and members of the press have been invited to join us as observers. This is not a news conference, however. We will not be entertaining questions from the audience. Reporters who would like to talk with members of the committee are kindly asked to touch base with the staff officer, Carol Laney, and also anyone who would like to submit a written comment or a document to the committee is welcome to do so by clicking the Provide Feedback link on the right side of the study website. You can also subscribe for updates about the study by signing up for the National Academy's Board on Agriculture and Natural Resources listserv, which is also to the right of the page. I would like to remind everybody that this is an information gathering session. That is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and coming to our recommendations. Therefore, I ask everyone to be extremely mindful that the committee has made no conclusions thus far, and that it would be a mistake for anyone to leave the meeting today thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee, and the committee must then respond to this review with appropriate revisions that will satisfy the Academy's Report Review Committee and the NAS president before it can be considered an official Academy's report. This committee is composed of 15 volunteer experts, and their names are listed on your screen. That'll be coming shortly. You can find a short biography of each member on the study website. I'd now like to turn things over to Carol Laney, a senior program officer with the Board on Agriculture and Natural Resources at the National Academies, and the study director for this activity. Carol will give you a brief overview of the National Academy's study process that we're undergoing. Carol?
Thanks, Diana. The National Academy, I just want to give a brief overview of the National Academies for anybody who might not be familiar with the institution and joining us for the first time today. So the National Academies of Sciences was first given a charter underneath, signed into law by President Lincoln in 1863. And later, the Academy was also granted the authority to create the National Academy of Engineering in 1964 and the Institute of Medicine in 1970, which today is known as the National Academy of Medicine. Collectively, we're known as the National Academies of Sciences, Engineering, and Medicine. And in addition to the 3 honorific bodies, we also carry out the work of scientists across the country, both inside and outside of these societies, to provide expert advice to the nation involving issues of science, engineering, and medicine. The mission detailed in the charter of the National Academies is to investigate, examine, and experiment upon any subject of art or science whenever called upon by the government. We fulfilled this mission through many convening activities like this consensus study that we're gathered for today. Just to share a little bit more detail about consensus study, as opposed to other convening activities that we do, such as workshops and forums, consensus studies are conducted by committees convened specifically for a unique statement of task. The experts who serve on these committees do so as individuals, not as representatives of any organization or institution to which they may belong. They also serve as volunteers. Consensus study committees review the state of scientific literature that corresponds with their task, and they write a report that will hopefully be used to shape sound policies, inform public opinion, and advance the pursuit of the scientific field in question. Each consensus study goes through a number of stages. So first, the sponsor and the National Academies work out what the statement of task will be. And the sponsor for this study is USDA's National Institute of Food and Agriculture. Then the National Academy solicits nominations to identify individuals with the necessary expertise and perspectives that can address the statement of task and contains a broad points of view. Nominees are appointed to the committee by the president of the National Academy of Sciences, Dr. Marcia McNutt, and their committee membership is provisional until they have completed a discussion of conflict of interest, disclosure, and composition at one of their early meetings. After being appointed, the committee moves into the information gathering phase, which is the phase of the study that we're in right now. They invite in speakers to speak with them in person or on Zoom. They may also look at information that's been submitted by the public. And then they begin deliberating on their statement of task. After they've started some deliberations, they begin working on a draft report. And when the draft report is complete, it's submitted to the National Academy's Report Review Committee. In that part of the process, a group of peer reviewers are recruited with similar expertise as to those serving on the committee to provide constructive comments on the draft report. The committee revises the report in response to those comments and then submits the revised report back to the Report Review Committee, which decides whether the report has been adequately, whether the revised report adequately addresses the reviewer comments. When the Report Review Committee believes that the report is ready, then it's released to the sponsor and made publicly available. I just want to say a few more words about the committee process. When the committee hears from speakers, as we're doing today, we meet in open session. This means that not only are speakers in the committee present, but anyone may be present, either watching if we're meeting in person, then in the room if they'd like, or watching online. When the committee meets in closed session to deliberate on its statement of task and devise the best way to address the report and the statement of task, then we meet in closed session when only committee members and National Academy staff are present. This is an overview of the timeline for this study. Today is our 7th public meeting. We plan to hold 3 more public meetings in July, including an in-person meeting in DC on July 26th. Information about those upcoming sessions are available on the study's website. I anticipate that the draft report for this statement of task will be ready for review in the fall, and that the final report will be released in the spring of 2024. If you haven't seen it already, here's the committee's statement of task, and I'll just give you a moment to look over it. And Kat, if you'd go to the next slide. As I mentioned, USDA's National Institute of Food and Agriculture is the sponsor for this study, and the study came to the National Academies via USDA from a congressional request in the last appropriations legislation. Finally, here's a screenshot of the study's website. On the left, you can see the tabs to navigate to the full statement of task and to the committee membership roster, which includes bios of each member. On the right, there's a link to provide feedback or comments on the study and a place to subscribe for updates about the study. If you need to contact the National Academy staff for anything related to this study, you have my email address here, as well as that of my colleague, Catherine Kane. Now, with that, I will pass it back to you, Diana.
Okay, thank you. It's now my pleasure to introduce today's speaker. Martin Blaser holds the Henry Rutgers chair of the human microbiome at Rutgers University, and serves as director of the Center for Advanced Biotechnology and Medicine. Previously, he served as chair of the Department of Medicine at New York University. A physician and a microbiologist, Dr. Blaser has been studying the relationships we have with the human microbiome, the bacteria that live in us and on us. Over the past 20 years, he has also been actively studying the relationship of the human microbiome with health and important diseases such as asthma, obesity, diabetes, and cancer. He has served as president of the Infectious Diseases Society of America, chair of the board of scientific counselors of the National Cancer Institute, chair of the advisory board for clinical research of the NIH, and until recently, chair of the Presidential Advisory Council for combating antibiotic-resistant bacteria under 3 presidential administrations. Dr. Blaser, thank you for joining us today. It's a real honor to have you.
All right. Thank you very much for your invitation. Are you able to hear me?
Yes, yes.
Okay, I'm going to share my screen then. Let's see now, I'm going to just make this smaller. Can you see my screen?
Yes, I can see the whole slide presentation. I mean, the whole slide, yes.
Okay, great. So I'll begin. Dr. Wall, thank you again, and thank you to your panel. This was an unusual invitation for me because, as you'll learn, I know little about soil, but I've been very interested in ecological changes in the human microbiome, and I believe that it is parallel to phenomena going on in the soil. So at the risk of boring you, I will tell you about our work on the human microbiome. The title of my talk is Antibiotic Perturbation of the Human Microbiome and Its Consequences, a Model to Understand Pressures on Soil. So in this slide, we're looking at several different weather phenomena, a hurricane, a drought, another hurricane, drying, warming of the polar regions. These might be 5 independent events that happened at different times in different places. And so we wonder what caused these. I think we all know that these are all caused by the same thing. They are a manifestation of a global process that we call climate change or global warming. And we could define that as a change in our macroecology. And what I want to talk about today is a change in our microecology, the microecology within the human body, and one might think about the microecology of the soil as well as a kind of global event. So in particular, I've been concerned about why have many diseases increased in recent decades. And this slide shows some examples. Diseases of the esophagus going up, juvenile diabetes doubling every 20 or 25 years, asthma hitting the roof. And to that, we could add obesity, inflammatory bowel disease, celiac disease, food allergies, atopy, attention deficit disorder, autism. If 10 diseases are going up more or less at the same time, do they have 10 different causes? Or perhaps there's a single cause underlying them all. And that's really what I want to discuss today, the idea that a change in the human microbiome, a single event, is underlying all these changes in human health. So let's start with obesity. This slide is obesity trends among US children and adolescents. The data are from the NHANES over about a 35-year period, looking at percentage of kids who are overweight. And even in the youngest age group, you can see which way the trends are going. Over time, the children of the United States are getting bigger. And we know that overweight children become overweight adolescents, become overweight adults. This slide looks at a comparison of time trends of overweight and obese children globally. So in developed countries like the United States, they're shown in this red line, and you can see the shape of the curve. Well, in fact, the same phenomena is going on in developing countries. It started lower, but is moving in the same direction. And if you see where the developing countries are today, it's where we were about 30 years ago. And so this is one piece of evidence that the same thing is going on around the world. So could the worsening pandemics that I've mentioned be related to changes in the human microbiome? Well, I want to give you a microbiologist view of the human body. So here's the human body, the number of cells in the human body. 3 times 10 to the 13th, those are human cells. 4 times 10 to the 13th, these are the microbial cells. It raises the interesting question, who are we? Here's a geneticist view of the human body. Unique human genes, 23,000. Unique bacterial genes in the average person, about 2 million. About 99% of our genes are microbial, are unique genes. So we might ask, what are they doing? We could ask several biological questions related to the human microbiome. What's the identity of the microbes that populate the host? What are they doing? How is the host responding to them? What are the forces that maintain equilibrium among the populations? What are the unique characteristics of each individual? And also, how can we manipulate it? And my guess is that you could ask very similar questions about the microbes of the soil as well. So let's start to think about the microbes in the human body. This slide is from a study done by Nick Boklich, who was a postdoctoral fellow in my lab. He studied 42 children in New York. And on this graph is the composition of their microbiome by month in the first 2 years of life. And the y-axis is the relative abundance of particular taxa. Now, the names of the taxa aren't that important. What's important are the colors. So we see that we begin life with this purple taxa, which we used to call the proteobacteria. And then these are gradually declining, and they're being replaced by bifidobacteria, Bacteroides, Clostridium, Faecalibacter. This is not a random event. This is quite ordered. You can look at children all over the world and you will see essentially the same choreography. This is an ecological succession. So where do these come from? Well, many of them come from mom. We humans are microbes. We humans are mammals. And we begin life in a womb that is sterile or mostly sterile. And then when we descend through the birth canal, when the water breaks, we are covered by mom's microbes. We swallow the microbes. Now there's skin-to-skin contact of infant and mom. The baby's mouth full of microbes inoculates the breast. And now chemical substrate and microbes go in to form the foundation of the microbiota of the gastrointestinal tract. Moms are kissing babies, licking babies, pre-masticating food. Lots of redundant ways for the intergenerational transfer of microbes from mothers to children. This is the way it has always been, not just in humans, but in all mammals, and in fact, essentially in all animals as well. But now, moms are different than they used to be. They live in a world of antisepsis, of antibiotics, of dietary antimicrobials. And babies are different too. They may be born by C-section, missing the journey through the birth canal. Today in the United States, 1 baby out of 3 is born by C-section. In some countries, it's 1 baby out of 2. Babies are bathed extensively. They're given formula, a chemical composition that is similar to, but not the same as human milk. And they're getting a lot of exposure to drugs, especially antibiotics, which will be the focus of my talk today. So over the last 20 years, I and colleagues have developed a concept called the theory of the disappearing microbiota. And this theory has 2 major tenets. The first is a changed human ecology by altering transmission and maintenance of ancestral microbes affects the composition of the microbiota, and that the microbes usually acquired early in life are especially important since they affect a developmentally critical stage. Now, about 15 years ago, Stan Falco and I enlarged this hypothesis, which is shown here, the effect of maternal status on the resident microbiota of the next generation. Our idea was that ancient moms would give their babies a replete ancient microbiota. But if they lost microbes and couldn't get them back horizontally, then the next generation would be born at a deficit, and so on and so forth. So in this model, the change of the microbiota at each generation is cumulative. Each one is starting at a lower point. This is our hypothesis for what has been going on in human populations, especially in the United States, in the 20th century and now into the 21st century. In fact, there is a growing body of evidence that this hypothesis is correct. I'll show you a few pieces of evidence. Here we're going to look at comparisons of the intestinal microbiota in 4 populations. This is work that Jose Clemente did with Maria Gloria Dominguez, Rob Knight, and myself. We look at 4 different groups of children: children in the US, children in villages in Africa and Latin America, and an uncontacted group of Amerindians. And here in this graph is a measurement of the amount of diversity in their microbiota. We have the least, the uncontacted children have the most. And you can imagine the same kind of step that we're talking about. Here from the same study, we're looking at an indicator organism, an organism Oxalobacter formigenes, which is related to kidney stones, highest prevalence and abundance in the uncontacted Amerindians, lowest in the US, intermediate in these others. And there has been much evidence supporting this. Here's a paper that just appeared this month in Cell, looking at species richness in 4 human populations, not the same populations that we studied. And here's California. The average person has 277 total species in their gut, but the most are the hunter-gatherer Hadza, 730, and 2 different communities in Nepal are intermediate. All of this is evidence consistent with the idea that we have been losing microbial diversity over the course of not hundreds of years, but over generations. So what are the consequences? Well, that means that there is loss of contingency species and genes. What are contingency species? These are species and genes that can have effect on host responses to environmental exposures that are toxic, metabolic, or infectious. Perhaps something similar is going on in soil. So what's causing this? In fact, there are many causes. I'm just going to focus on one group of chemicals that are playing a role, and this is antibiotics. We all recognize antibiotics as among the greatest discoveries of the 20th century. Here's Fleming reenacting the discovery of penicillin. Antibiotics have been in the practice since the late 1940s. They've saved innumerable lives. They've revolutionized every area of medicine. And perhaps you have chemical products that you could say the same in agriculture. And as a result, practitioners are using them more and more and more. How much more? Well, here are some scales of antibiotic exposure. Recent estimate, 73 billion antibiotic doses used worldwide on an annual basis. That's 10 antibiotic pills for every man, woman, and child on Earth, and the numbers are increasing. In the US, the CDC has counted courses of antibiotics. The average child, by the time they're 2, has gotten 3 courses. By the time they're 10, 10 courses. Pregnant women, just before the intergenerational handoff, more than 50% exposed to antibiotics. There's tremendous variation in use, which I'll show you. And there are also exposures from antibiotic use on the farm. We don't even know the magnitude of this for humans. Now, I mentioned about variation. This slide comes from CDC data looking at 250 million outpatient antibiotic prescriptions in the US in 2018. And the states are color-coded according to their antibiotic prescriptions per capita. And what we could see is that in this group of dark red states, antibiotic use is more than double the use in these light states. Now, this is the same country, and there's tremendous variation. These phenomena are stable over many years. And this does not reflect differences in serious bacterial infections. This reflects differences in the practice of medicine, in the culture of medicine. Here's per capita antibiotic prescribing in 31 European countries. From the Netherlands, the least, to Greece, the most, there's more than a 3-fold difference in antibiotic prescribing per capita. There is not a comparable difference in the rate of serious bacterial infections. Here's work in 8 cohorts in developed countries, in low and middle income countries, work supported by the Bill and Melinda Gates Foundations. Here we're looking at children in the first 2 years of life, antibiotic courses per person year. I've drawn in the line for the US. What you can see is that in 6 of the 8 countries, the use is greater than in the US, including in this cohort in Bangladesh and in Pakistan, where the average child is getting more than 10 courses in the first year of life. Now, you might ask, how is that possible? But it turns out that these children have parents who love them. The child has a fever or a cough, and they go to the local pharmacist or street vendor who is happy to sell them an antibiotic. So we're concerned about the ecological effects of antibiotic exposure, and I draw these like the proverbial iceberg. The tip of the iceberg, the part that we can see, is antibiotic resistance. We've known about this at least since 1945, since Fleming gave his Nobel address. But I believe that the body of the iceberg, like the iceberg, the bigger part of the iceberg, below the water, silent, is the effects of antibiotics disrupting the microbiome, leading to clinical consequences. These disruptions could be transient or long-term. The effects could be developmental, situational, senescent, or generational. And the manifestations could be immunologic, metabolic, neoplastic, or maternal. There is evidence for everything on this slide. I'm just going to show you a few examples in the time that we have. But perhaps something similar to this is going on in the soil. So the first question is, does early life antibiotic exposure correlate with subsequent disease risk? Is there any linkage at all? So first, I'd like to show you a study that I conducted with colleagues at the Mayo Clinic, assessing associations of early life antibiotic exposure with subsequent health problems in children. This was done in Olmsted County, Minnesota, where the Mayo Clinic is located. It's a very stable population. We were able to find 18,000 children who were born in about a 13-year period, and we could analyze about 14,000 of the children. We were interested in two periods, a period of antibiotic exposure before the age of 2 and health outcomes after the age of 2. And about 10,000 children were exposed before 2, 4,000 not. They were exposed to the 3 most common antibiotics used in childhood. So we looked at 10 different health conditions. Here, I'm going to show you a Kaplan-Meier plot of the time to developing asthma by sex and by antibiotic exposure. So on the x-axis is the age from 2. This is the outcome period up to the age of 14. The y-axis is the probability of asthma. The red lines are girls, the blue lines are boys. The solid red line is the rate of asthma development in the girls who did not have early life antibiotic exposure, and the dotted line is girls who did. Blue line, boys without antibiotic exposure, first 2 years of life. Dotted line with exposure. You can see a marked increase in both of these groups. As I told you, we looked at 10 different health conditions. Inflammatory and allergic conditions like asthma, hay fever, food allergy, atopic dermatitis, celiac disease, metabolic abnormalities, overweight and obesity, neurodevelopmental abnormalities, ADHD, autism, learning disability. Here's the hazard ratio. A hazard ratio of 1 is neutral, no relationship between antibiotics and the health condition. But in fact, for all 10, the hazard ratio is greater than 1. And in 8 of the 10, the differences are statistically significant. In further analysis, and you can see it in the paper, we found specific associations with number of antibiotic courses, a dose response, the timing of exposure during the first 2 years of life, and antibiotic class. So that work suggests there could be a linkage, but that's correlation. So the question is, do antibiotic-induced perturbations cause the clinical conditions? Well, to address this question, we first considered a paradigm, and that is the use of antibiotics on the farm. More than almost 80 years ago, farmers discovered that if they used antibiotics in their livestock, they could promote their growth. As you, I'm sure, are well aware, this is what's called growth promotion. And it works from chickens to cows, a wide swath of vertebrate evolution. Importantly, this box here shows a list of representative antibacterials. It works with all of them, regardless of their chemical structure, their class, their target, or their spectrum. Antivirals do not work, antifungals do not work. It's specifically antibacterials. And here is the effect of timing. This is the effect in swine on growth rate. The earlier the antibiotic is started, the bigger the effect on growth, the bigger the effect on feed efficiency, the conversion of food calories into body mass. This is what farmers are trying to do. And this suggested to us a developmental phenomenon. So we began to do studies in mice in which we would give mice antibiotics or not, examine properties, analyze the microbiome, and examine for relationship. We have done many studies. I'm just going to show you a few that I think are illustrative. So the first study was done by Dr. Il-Sung Cho when he was a postdoctoral fellow in my lab. Il-Sung gave mice 4 different regimens of antibiotics at the midpoint of the FDA-approved level on the farm, or no antibiotics. And then he measured percent body fat in the mice after a number of weeks. And what he found is that all 4 antibiotic regimens increased body fat percentage compared to the control. You can see an illustration of this. This was our first evidence that antibiotics were changing metabolism in mice. Now, Il-Sung showed many other things, but I want to proceed to the next study, which was done by Laurie Cox when she was a PhD student in the lab. Laurie wanted to know what was the effect of combining a diet rich in calories, a high-fat diet, and antibiotics. So she gave mice the low dose of penicillin that as used on the farm, or not. She had all the mice initially on normal chow, and at week 17, she switched half of the mice to a high-fat diet. Here's the major result in morphometry. She studied male mice and female mice, the total mass of the mice, their fat mass, and their lean mass. If we start with the male mice, this black line is the control group. No antibiotics, normal chow. Normal chow plus antibiotics, they're bigger, just like on the farm. High-fat diet, bigger still. High-fat plus antibiotics, biggest. Lean mass, increased on antibiotics, just like on the farm. Fat mass really increases on high-fat diet, and even more high-fat plus antibiotics. Female mice, many of the same effects. If we look at fat mass, the average female mouse on the high-fat diet gained 5 grams of body fat, but high-fat diet plus antibiotics, 10 grams. They doubled the amount of body fat. The antibiotic potentiated the effect of the high-fat diet. Laurie showed many changes in the liver that are consistent with this that you can read in the paper. Now, up to this point, the mice were getting antibiotics for life, but that's not what children get. So Laurie did an experiment that we called Durastat. She asked, are the changes durable if the antibiotic exposure is only limited? So in addition to a group of mice that did not receive antibiotics or got lifetime antibiotics, she had mice that received only 8 weeks or only 4 weeks of antibiotics. Here's the effect on morphometry in female mice, total mass, lean mass, and fat mass. The black line is the control group. What Laurie found is that all 3 groups of antibiotic exposures led to increased total, lean, and fat mass. So 4 weeks at the beginning of life was sufficient for the full effect. We did another experiment where we started later. There was an effect, but not as strong. This was the critical window. We were interested in the effects on the immune responses in the mice. And here I'm showing work by Jackie Leung and Peng Lok's lab, looking at the effect of the subtherapeutic antibiotics on Th17 populations in the intestine. This is an important group of T helper lymphocytes in homeostasis. No matter how we looked at Th17 in the small intestine or the large intestine, it was always down in the group that received the antibiotics. So now what's going on with the microbiome? So here we're looking at the fecal community structure in the intestine in mice that are 3 weeks old. And at 3 weeks, there are only 2 groups of mice. The mice in the control group, no antibiotic exposure, or these other 3 groups, they're still all getting antibiotics. So here's a principal coordinates analysis. The black dots, circles are the control mice. The orange are the antibiotic mice. There's a lot of variation here. You can see that they're largely overlapping, but there's some difference. Not surprising, one group is getting antibiotics. Now we're going to look at 8 weeks. And at 8 weeks, there are 3 groups of mice. Control mice, still no antibiotics, continued antibiotics, or one group of mice got antibiotics for 4 weeks and then it stopped. So here's their microbiota at 8 weeks, control in black, antibiotic in orange, more separate than before, not surprising. They received more antibiotics. But the group that got antibiotics and stopped, their microbiota normalized. So that meant that the effect of the antibiotic was transient, but the effect on the phenotype was permanent. And this was our first evidence that altering the microbiome at a critical early stage in development could have long-term consequences. And we've confirmed this with other studies. So was this an antibiotic effect or was this a microbiome effect? So to answer that question, we did a transfer experiment. We gave mice antibiotics or not. We sacrificed them and harvested the microbiota in their cecum, and we gave them to germ-free mice and followed them for 5 weeks. These mice never saw an antibiotic. All they saw was a perturbed microbiota or a normal microbiota. Here's the effect over the 5 weeks on total lean and fat mass. The black line is the control group. The mice that received the perturbed microbiota had increased total mass, no effect on lean mass, and an increased effect on fat mass. So the perturbed microbiota were sufficient for the metabolic signal. The signal is in the perturbed microbes. Something might be similar in plants. Now we were interested in immune phenomena. So we're looking at expression of genes involved in intestinal defenses in the donor mice. So here we're looking at Th17, the transcription factor for Th17, 2 cytokines downstream of Th17, 2 antimicrobial peptides affected by Th17. As we came to expect, compared to the control mice, the mice that directly received the antibiotics had low levels in all of these genes. Now we're going to ask what happens when we transfer the microbiota from these mice to the germ-free recipient mice. And we see that we recapitulate the phenotype. Again, for this phenotype, the perturbed microbiota was sufficient for the immunological effect. So here we show in this experiment both metabolic and immunologic phenotypes from the perturbed microbiota. Now, these are models. So we wanted to do a little work on disease. So Shusang Zhang, a postdoctoral scholar in the lab, looked at the effect of antibiotic courses on a mouse model of type 1 diabetes, juvenile diabetes, in the NOD mouse. This is a mouse that spontaneously develops these autoimmune form of diabetes. Instead of continuous low-dose antibiotics, he gave mice pulses of antibiotics as if he were treating their ear infections or their throat infections. So he gave mice either 3 courses of antibiotics or not, or 1 course of antibiotics or not, and then he measured diabetes. And this is a very nice model because they develop diabetes on average 20 weeks after birth. So there's a big window of latency. So here's the Kaplan-Meier analysis of type 1 variants. We're looking at male mice here. The blue lines are the control mice. They did not receive antibiotics. They're developing type 1 as expected of male NOD mice. But the male NOD mice that got the antibiotics, they're developing diabetes earlier and more of it. So antibiotics are driving this phenotype. They're driving this disease. We looked at many different aspects. Again, you can look at the paper. I want to look at the effect of the altered microbiome, the antibiotic altered microbiome on gene expression in the ileum, in the small intestine of the mice. Here we're looking at global gene expression patterns in mice that are sequentially older, and you can see that there is a maturational pathway. Here, we know what all the genes are here. We can put the genes into different patterns. We can say here are the genes that matured in the control, whose expression matured in the control mice. These are the ones that matured in the antibiotic mice. These are the ones that matured in both. So the maturation was resistant to antibiotics. These just matured in the controls, not in the antibiotics, so they were affected by the antibiotics. And here's a group that were dismatured. As I said, we know all the genes. We could put them into pathways. And here we're looking at differential toll-like receptor signaling pathway expression in the ileal wall. This schematic shows all the genes in the TLR system. All the genes in color are genes that are affected, either increased or decreased by the antibiotic exposure. So there's a big effect on signal transduction in the mouse. Final study I want to show here, it has to do with inflammatory bowel disease. There's an epidemiologic study in Denmark of all the children in Denmark. They have very good records of antibiotic exposure. And so they asked, is there any relationship between the number of courses that children are getting early in life and the development of IBD? And what they found is yes, more courses of antibiotics, more likely that a child would develop IBD, especially Crohn's disease. So we wanted to study this in a mouse model to ask, can an antibiotic altered microbiota affect IBD outcome? And to make it more interesting, we wanted to look in the next generation. So we studied wild type mice or IL-10 deficient mice. These are mice that spontaneously develop colitis. This work was done by Angel Schulfer when she was a graduate student in the lab. She gave mice control normal microbiota or antibiotic perturbed microbiota. She gave this to germ-free mice that were pregnant. So she had wild type or IL-10 deficient mice, 2 genotypes, 2 inoculars. She had 4 groups of mice. She inoculated the mice. The pregnant mice soon gave birth to their offspring. And then we followed the offspring until they were middle-aged. We were interested in the effects on the ecology of the gut and the effects on disease. There were many effects on the ecology. I can refer you to the paper. We're going to look at the effects on the disease, the disease phenotype, especially in the IL-10 mice. These are the mice that develop colitis. So here we're looking at colonic pathology in the IL-10 pups at week 21, according to the microbes to which their mothers were exposed. Here's the colon of a control mouse, of a mouse whose mother got the control, the normal microbiota. This mouse has colitis. We expect that in an IL-10 knockout mouse at 21 weeks. But this is the colon of a mouse whose mother got the antibiotic perturbed microbiota. They have a tremendous amount of colitis. In fact, the difference is 30-fold. This is not a trivial difference. So there's a very strong phenotype. So if I had to summarize this experiment, what I would remind you is that neither the pups nor their mother ever received an antibiotic. They never saw an antibiotic. That meant that the enhanced disease signal is entirely microbial in the founding inoculum. It also means that antibiotic effects can cross generations. And it also implies that inheritance is not only based on host genes, but it's based on microbes and their genes as well. So let me just summarize what I've said so far. I've shown evidence that antibiotics have long-term effects on host metabolic and immune development. The effects are due to perturbing the microbiome. Other factors of modern life also contribute. Effects may be transmitted to the next generation. We need to find and implement solutions. I haven't discussed other aspects of modern life, just using antibiotics as the model. But I'm speaking to people who are interested in farms, and you know that there are a lot of exposures of the soil, including pesticides, antibiotics, microplastics, etc. So if we think about the threats to the microecology of soil and humans, we share a lot of threats: antibiotics, antifungals, pesticides, microplastics, heavy metals. In the soil, there are threats from fertilizers. In humans, we have C-sections and formula, but there's a lot of parallels. So up to this point, I've mentioned a pretty bleak story, but if it's happening, we may as well understand what's happening. So where's the hope? Well, if we want to solve the problem in humans, we have to stop killing the beneficial bacteria, and the solutions involve curtailing unnecessary antibiotic use. I'm sure there are analogies on the farm. And then we have to think about how to restore the depleted organisms. So we have been working on that, and I'm going to show you one example going back to Shusong Zhang's studies on the type 1 diabetes model. Instead of 2 groups, we now have 3 groups of mice: control mice, no antibiotics, an antibiotic group, and a 3rd group that got antibiotics and then got a cecal transplant from mothers. This is cecal microbes after the antibiotic exposure. So what's the effect on the type 1 diabetes? Here's the Kaplan-Meier curve. Blue line is the control group. They're getting diabetes as expected. Red line is the antibiotic group. They're getting diabetes even more than control, as we now expected. And the green line is antibiotics plus mom poop, and we have been able to partially restore. We've almost come back to the baseline level. So this is a proof of principle that after the fact, we can do a restoration. Now I'll show you one other result from this slide. We're looking at differential ileal gene expression in relation to the restoration. So this is a heat map based on RNA-seq data. Each column is one mouse. Each row is one gene. And here we have an unsupervised hierarchical clustering. The mice that receive the antibiotics cluster by themselves. The control mice cluster here. The mice who received antibiotics and got the mom poop, their gene expression is now clustering with the controls largely, not clustering with the antibiotics, even though that's what they received. So in addition to this proof of principle, this is a pathway for discovery, to discover the microbes, the microbial genes, the metabolites, the host genes perturbed by antibiotics that drive autoimmunity and that ultimately can be restored. So what's the future going to bring? Here was our picture of diversity loss of the microbiome in 3 model locales. This blue line would be the United States, maybe 7 generations into modernization, depleting cumulatively. Red could be India or China, starting late, but catching up in a hurry. Yellow might be countries in Africa or Latin America have just begun on this depletion process. This was our view in 2016. The question is, what's the future going to bring? Is the microbiome going to decline further? Are we going to be able to arrest the decline? Or maybe our next generation is going to be able to reverse it through restorative steps. It may not be that simple because now opportunistic organisms have come in, taking up these niches, and we may have to remove them before we restore. Finally, what's the medicine of the future going to look like? My view is that pediatricians of the future will examine newborn babies and they'll examine their diapers. And they'll ask, does this baby have the global microbes that all babies should have? Do they have the personal microbes that a baby of a particular genotype should have? And if they don't, they will reach into their new armamentarium and administer these microbes and ultimately reshape the microbiome as needed to optimize their health trajectory using a combination of diet, prebiotics, probiotics, and symbiotics. Now, the microbiome is disappearing. Where are they going to get these organisms? And I want to tell you about one last initiative called the Microbiota Vault. It's very parallel to the Seed Vault. In fact, it's inspired by the Seed Vault, a global nonprofit to conserve long-term health for humanity. We want to store the microbiome, especially from developing countries, before it is lost. You can read about it in our paper of science. This is led by Maria Gloria Dominguez. You can go to our website, www.microbiotavault.org. Finally, I just want to tell you that a film has been made about this whole area by independent filmmakers, Sarah Shank and Steve Lawrence. It's called The Invisible Extinction. It's now available, it's now available on Amazon, Apple, iTunes, and Vimeo. It is a film for the general public, so they understand the issues that we're talking about this extinction. But it's been well received in the scientific community. And with that, I will stop, and I'm happy to answer your questions.
Thank you so much, Martin. That was really both stimulating, interesting, and scary a little bit. Already there are hands appearing from the committee. Sam, can we see you also? Oh, you're clapping. I see. He's clapping for your presentation. Are there questions from the committee? Lots of claps.
I have a question.
Sure, Kate.
I'm Kate Scow, soil microbial ecologist, so on the soil side of things. And one really interesting analogy between human microbiome and soil microbiome is this whole idea of pro and prebiotics. And in soil, we often think about diversity in soil and, do you need to inoculate with new organisms, or can you restore it by, providing the right kinds of substrates or even just feeding the soil? which we often don't do because we're thinking only about the plant. And is there an analogy with the human microbiome? And I'm thinking also of their ability to respond to antibiotics.
Yeah.
You know,
So
the prebiotic important part of that.
I'm very familiar with pre and probiotics in humans. I'm not familiar in soil, but I can tell you that there are bacterial cultures that are being sold all over the world by the tens of millions called probiotics. These are microbes, sometimes of human origin, sometimes not. They're often alive, sometimes they're dead. They are generally considered as safe. They're grass. They're almost completely unregulated, and they're almost completely untested. And millions of consumers are taking these as an act of faith that this will improve them. And in some cases, it does. The FDA doesn't allow actual health claims, but they have vague health claims, and it's a multi-billion-dollar industry. It's an industry that's driven almost entirely on marketing. However, I think that probiotics are going to be really important in the future, but we're going to have to define them scientifically. And it may be that your probiotic is different from mine. And on the farm, all different soil types, all different plant types, I would imagine that there will be a diversity of probiotics that can be given. But I think probiotics are kind of people trying to short circuit the thing. They think that it's a restoration, but it's not really a restoration. These organisms don't persist. And if we've lost 400 different species, taking one or two that don't persist, I'm not sure how that's going to help us. So that's the short of the answer. I think there's a lot of science that has to be done both in humans and on the farm, so that we can develop responses to this extinction crisis.
Thank you.
Got a couple more hands here. Sam?
Thank you very much, Dr. Blaser. It was a fascinating talk. Really enjoyed and also enjoyed your papers on disappearing microbiota and other papers. So I was looking at the biodiversity loss and how antibiotic use has kind of further worsened that the biodiversity loss in the gut microbiota. I was wondering the effects, that phenotype effects and other effects that you have observed in your studies, to what extent or to what proportion that could be attributed to the overall alpha diversity loss or biodiversity loss and the loss of key functional groups. Is it because of the overall loss, just because when there is more diversity, there is less niche available for potential pathogens perhaps, or it's the key functional, some of those important functional groups that are lost in this process that is kind of causing, and that could be attributed to these, there's some of these phenotypic effects.
Yeah. That's a wonderful question. And it's really a question at the heart of the field. The short answer is that so many things are happening simultaneously that we really need to understand what are the key elements. And the loss of diversity, I think is indicative, and that's why I mentioned this idea of the loss of contingency genes. We're just losing tremendous richness. But if we go back to some of my earlier slides, the slide, there are 2 points I want to make. One is they figured it out already on the farm. Antibiotics work across animal species. It's not one species. It's affecting a very conserved element in the biology. We'll just make it simple, in mammalian biology. And young animals are programmed. They have to develop their metabolic programs, and they basically have one decision to make. How much energy do you save and how much do you spend? And our work suggests that this is being put out of balance by the change in the microbial composition through such things as short chain fatty acids and effects on liver and adipose tissue. That's where some of our work is going. again, and the really key point that I think is important is early life. That's when everything is developing. That's when the trajectory is developing. That's when metabolism, immunity, and cognition, they're all developing in parallel in young kids. That's the most vulnerable window. So we and others are working on different mechanisms, and there are many different mechanisms. This is, I just wanted to give you the overview. And I think that it has to do very much with the change in composition and how it is interfacing, for example, with innate immunity and with energy efficiency.
Thank you.
Carlotta. Can't hear you. I don't know what's going on.
Can you hear me now?
Yes.
Oh, okay. Sorry about that. Thank you very much for this fascinating talk, Dr. Blaser, and for sharing this very important work with us. My question goes back to your earlier point about restoration of the gut microbiome and the human microbiome in general. And we know from fecal transplant studies in humans that many of these changes are not long lasting and may be very dependent on what human beings do and what's in their environment. So thinking about restoring microbial diversity at a large scale, have you thought of strategies or solutions that we can think of to make those changes that may be induced by transplantation or pre and probiotics long lasting if our environment continues changing and our diets and our behaviors continue changing?
Yes, that's a wonderful question also, and we could spend the next hour discussing it. I'm just going to highlight one feature, and that is that in my own work, I'm really concerned about children. I'm really concerned about the next generation. I'm not actively working on people who already have disease, of which there are many. I'm really interested in preventing disease in our children and our grandchildren, in the next generations, because I think it's stepping down by generations. So if we don't act, it's just going to get worse. So part of our work is to understand what are some of the missing microbes, some of the microbes that have disappeared in early life, and thinking about them as restoratives. So in my dream scenario, we will identify 4 or 5 really key organisms that we will give to young children who don't have them, maybe in the first 6 months of life, just like we vaccinate kids early in life to prevent something. This is a prevention also. It's not a vaccination, it's an inoculation. And we already have some candidate organisms that we may be able to give back. For example, in kids who are at high risk for type 1 diabetes, in type 1 diabetes families. I would start obviously with the most high risk kids, high risk for asthma. And we and others have identified certain constellations of organisms that, at least in the mouse, appear protective. So that's one place to start, which is to name the organisms. Another is to make engineered strains that have important functions that we want to, important metabolic or immunological interactive functions. But I think that since we lost natural organisms, since we lost our ancestral organisms, my goal is first to replenish those ancestral organisms. That's part of the rationale for the microbiota vault as well.
Thank you very much.
Hi, Mike, you're next.
Thank you. Along with the others, yes, my thanks as well for a really interesting presentation. I want to get back to this, your theory of sort of declining microbiota diversity along the generations. And I'm wondering, as you showed in children, there's changes in the microbiome or microbiomes, different places in the body over time. I mean, we all go through transitions in life. So later on, I presume there's changes going on with our diet and environment and so forth. I'm curious if you've looked at children's microbiome coming, derived with mothers at different ages. And I'm wondering, is there a link between the age of the mother and, changing in her microbiome over time? And, I don't know the statistics, but I presume that in the US, the age of motherhood is advancing. And so I'm just wondering if there's a link between age of mothers in this.
We haven't looked at that. Well, actually, maybe we did in that paper in Science Translational Medicine. But there seem to be three key factors. Maternal age, by and large, if it is a factor, it's not a big factor, and because usually these are relatively young women. The three big factors are C-section, whether a baby is born vaginally or by C-section. There's big difference in the microbiome. The second is antibiotics, both during pregnancy and in the first months of life, especially the first 6 months of life. And then breastfeeding versus formula. And the good news is that breastfeeding is kind of an antidote. It corrects many of this of we'll call them the sins, because breastfeeding is not accidental. The micronutrients in breast milk have evolved over 100 million years, specific for each mammal. There's conserved, but it is different. And breast milk is designed to nourish the beneficial microbiota. It has sugars that are indigestible by the baby. They are only digestible by certain members of the microbiota. Breast milk has urea in breast milk, a waste product of humans is in breast milk, because it is a nitrogen source for certain organisms that can utilize it, that doesn't compete for amino acids with the baby. I mean, nature is infinitely clever. So those are the three most important aspects: C-section, antibiotics, and on the negative side, formula feeding; on the positive side, breast milk.
Okay, do we have any more questions? Sam?
May I ask one more question, if you don't mind?
Sure.
Sorry, while I have you. That's a great answer. I mean, I think I think it's fascinating how breast milk has so many other micronutrients and so on. My question was, one of the slides that you showed, and you mentioned about prebiotics, probiotics, and symbiotics. Now, I'm familiar with prebiotics and probiotics, and I'm also familiar with syntrophy. What I don't know what is, that's a naive question. What is symbiotics?
The symbiotics, that term means kind of the coordinated effort of prebiotics and probiotics together, so that you give a probiotic and you give them the chemical nutrient that will encourage their growth. That's how the term symbiotic is used.
Mm-hmm. Has it been shown that when you use a combination of those two, it has a facilitative effects? It has a
yes. There's been extensive work. I don't know in humans, but certainly in animals that show that, and it stands to reason. If you give an organism and you give them a food that they will preferentially use that many other organisms cannot use, you give them a growth advantage. Remember, the introduced organism has to compete with all the organisms that are present, and that's one of the reasons. So if you take if you go to the health food store tonight and you take a probiotic, you will find that probiotic in your stool tomorrow and maybe the next day, but not beyond that. It will not succeed against the normal microbiota. That's good for the probiotic manufacturers. You have to take it every day, but it indicates how that per se isn't a good approach. Now, I'm hopeful that over the next decades we will have much better phenotyping of people with illness and for health prevention, and we will be able to say, okay, this is the microbe you need to take to slim you down, or so that you won't grow at a very fast trajectory, or that this is good for your lymphocytes so that you're not going to be wheezing. That's my hope.
Thank you.
Any more follow-up questions?
I just want to make one other statement.
Sure.
And that is that I have focused here on antibiotics because of the scale of antibiotics globally, and kind of the timing of introduction of antibiotics and all these changes. And of course, antibiotics are designed to inhibit and kill bacteria. So everything fits, but it is not the only agent. And I worry about things like glyphosate and microplastics and many of the same things that you worry about, because our microbiota is under a chemical assault.
Any further questions? These are really important points that you're making, and I think the committee will consider how to draw any parallels, and even if it may be indirect, of what we know and what we don't know. Nick has a question.
Well, I'll take a shot. I'm not a microbiome person, but what I'm wondering is when we start looking at the effect of soil health on human health, if you can give us an idea of what we should be looking for, because the key will be which management practices in agriculture, how it changes the microbial or the microbiome in the soil. So do you have a short list of things we should look for?
Let me just start with the disclaimer that I know about as much about soil health as you know about human health, divided by 10. So you're not getting it from an expert, but you know, I would worry about depletion of micronutrients, of metals and other vitamins, for example, so that the foods that come from those plants grown in depleted soil may be deficient in certain nutrients. I don't know whether that is true or not, but that's certainly where I would look.
That helps.
Thanks, Nick, for that question. Any comments? Well, Martin, I want to thank you so much for speaking with us today and helping the committee, you know, think about this aspect. Everybody's clapping again.
Let me just tell you that I'm in Portugal at this moment teaching in a course on symbiosis. It's a course we have 35 PhD students from around the world, and we're teaching about symbiosis in animals, insects, fish, plants. It's a terrific course at the Institute Gulbenkian.
It sounds like fun. I wish I could sign up for it. But really seriously, thanks so much, because I really think this helps the committee think about its statement of task and address this from a different perspective, and especially with regard to the human microbiome, which you pointed out, you know about, and some of us don't know very much about. I want to thank everybody online who's joining us also. And the committee will hold its next webinar on July 17th. So be sure and check that on our website. It'll be Monday. And thank you, everybody from the committee, for being here again. Kara, do you have anything else to add? Okay. All right. Thanks so much, Martin. Once again, it was really great.
Hi, everybody. Wonderful to talk to you.